After investigating the behavior of the conjugates within a reducing environment, the cellular internalization of the conjugates was then monitored. So in 2A, we are looking at the cellular internalization of the P14LR peptide and the P14-CanC conjugate. A P14-CanC fluorescent conjugate was made in order to monitor the cellular uptake of it. So the cellular uptake was investigated within macrophages, in which the macrophages were incubated with either the peptide or the conjugate for 1 hour and then were analyzed by flow cytometry. They wanted to see how the overall charge of the conjugate played a role with cellular uptake. This is because when you add kanamycin to the P14LR peptide, it increases the charge from a +8 to a +12, which might affect mammalian cell accumulation efficiency because mammalian cells have an internal negative charge as compared to their external environment. The x-axis is showing the concentration of the peptides and concentrations used in the 3 different tests, and the y-axis is showing the arbitrary fluorescent units, which is showing us how much fluorescence is occurring within the macrophages. So the results of this show that both the P14LR peptide and the P14-CanC fluorescent conjugate show a concentration-dependent cell penetration within the macrophages. The kanamycin conjugate seemed to have an increased cellular uptake as compared to the P14LR, but we see that the difference is not that significant because these bars here are overlapping. So really no change in cellular uptake is seen when you increase the positive charge. So since we know that these conjugates are able to penetrate the macrophage cells, they wanted to see where they localize within the cells. So in order to investigate this, macrophages were exposed to 10 micromoles of the conjugate and then were observed after 1 hour using confocal microscopy. In 2B, DAPI stain was used to stain the DNA blue, and lysotracker stain was used to stain endosomes and lysosomes, and it stained them red. Endosomes are organelles used to deliver materials at the cell surface to different places within the cell, including lysosomes. Lysosomes are organelles that have digestive enzymes that can digest things such as bacteria and viruses. The P14-CanC conjugate was green under the confocal microscope. So we see here that the P14-CanC fluorescent conjugate localized in the cytosol, the nucleus, and in the endosomes. We know it's in the endosomes because of this yellow color here. It's yellow because the red from the lysotracker stain merged with the green from the fluorescent conjugate, causing the yellow color. So this is indeed showing that the conjugate was able to localize to the endosomes. In 2C, DAPI stain was used again and the conjugate was also green, but mitotracker stain was used to stain mitochondria red. So after staining everything and looking at it through the microscope, we don't see any yellow overlap. This means that the P14-CanC fluorescent conjugate was unable to localize to mitochondria within the macrophages. This is interesting because the P14LR peptide is known to localize to mitochondria within cells. This just shows the importance of having the reducible linker within the P14-CanS conjugates. So the P14LR peptide can dissociate from the kanamycin and localize the mitochondria. So after observing the localization of the P14-CanC fluorescent conjugate within macrophages, the antimicrobial activity of the P14-CanS and CanC conjugates were evaluated. The activity of these conjugates were then compared to the activity of P14LR peptide P14SH, kanamycin, and a 1-to-1 mixture of P14LR and kanamycin. The antibacterial activity was evaluated by comparing the minimum inhibitory concentration of each conjugate. The minimum inhibitory concentration, or MIC for short, is the lowest concentration of the drug needed that would inhibit bacterial growth. This was determined using a broth microdilution technique, which consists of a sterile tray with multiple plates that contain different levels, different concentrations of conjugates and the components, but a fixed number of bacteria in each plate. And then the MIC was determined by seeing the plate with the lowest concentration that inhibited bacterial growth. Results from doing this experiment are shown here in Table 1, and they show that the P14 CAN-S conjugate was had very low MIC values that range from 0.12 micromoles to 2 micromoles. When compared to P14LR, we see that the P14 CAN-S conjugate is 2 to 32 times more active than the P14LR peptide alone. Is also 2 to 32 times more active than the kanamycin drug by itself. The P14 CAN-S conjugate is also 2 to 16 times more potent than the P14 CAN-C. This further shows that the reducible linker provides a potent antibacterial activity. The antibiotic activity of P14 CAN-S against S. aureus and S. epidermidis is also important because these two microbes form biofilms. Biofilms are composed of extracellular polymeric substance, which is very hard to penetrate with drugs. This low MIC value of 1 for both of these shows that the P14 CAN-S was able to penetrate the biofilm and effectively target the pathogens. So after evaluating the antibacterial activity of the conjugates, the mechanism of which they kill the bacteria was investigated. P14LR peptide has been shown to target bacteria in a non-membrane lytic fashion, meaning that it does not disrupt or break the membrane as a means of killing the bacteria. The activity of P14 CAN-C and CAN-S was then investigated to see if they disrupted bacterial membranes. This was done by monitoring the release of an enzyme called beta-galactosidase from E. coli cells. This is an enzyme that is used to break down large sugar molecules into smaller monosaccharides. They then compare the activity. They then compare the activity of the conjugates to the activity of water and melittin, which both served as controls. Melittin is a peptide that has powerful cell lytic abilities. So the blue line is the beta-galactosidase activity after E. coli was treated with water. The green was the beta-galactosidase activity after E. coli was treated with P14 CAN-S at 5 times its MIC. Purple was for the P14 CAN-C at 5 times its MIC, and then the red was the beta-galactosidase activity after E. coli was treated with melittin at 5 times its MIC value. The x-axis just represents the time spent monitoring the beta-galactosidase activity. The beta-galactosidase activity itself was monitoring by adding a beta-galactosidase substrate to the treated cells. The absorbance of the cleavage product from the beta-galactosidase substrate was then monitored at a wavelength of 405 nanometers, as shown on the y-axis. The beta-galactosidase leaked out of the cells due to their membranes being lysed, then we would see a higher absorbance due to a greater number of cleavage product. So at even 5 times their MIC values, we see that there really is no beta-galactosidase activity, meaning that the membranes were not lysed, even after 1 hour treatment. The results of this test just go to show that lyse was not the mechanism of antibacterial. Lysis of the membrane was not its mechanism of antibacterial activity. So then in Figure 3, they wanted to see the ability of the P14 CAN-S and CAN-C to rid intracellular pathogens from macrophages. The intracellular pathogens used were S. enteriditis, B. abortus, S. flexneri, M. smegmatis, and M. tuberculosis. The activity of these conjugates was then compared to the individual components and a 1-to-1 mixture of P14LR and kanamycin. So the y-axis here is showing the percentage of bacterial growth within the macrophages. The concentrations of all these being tested was 10 micromolars. And then the macrophage cells were then treated with one of these bacteria. They were treated for 9 hours. After being treated with this bacteria, they were treated for 9 hours with one of these conjugates, or the individual components. The P14 CAN-S conjugate was able to lower S. enteriditis levels to 5%, B. abortus levels to 15%, S. flexneri levels to 15%. The P14 CAN-C conjugate, however, behaves similar to the 1-to-1 mixture of kanamycin and P14LR, showing the importance of the reducible linker on intracellular potency, which allows the antibiotics to separate and go their own ways within macrophages. In Figure 3b, the y-axis is the same, but the x-axis is slightly similar because they did not test the P14 CAN-C conjugate. The concentrations of the components in the conjugate used, however, were also tested at 5 micromolars and 10 micromolars instead of just 10 micromolars up here. Both of these bacteria are part of Mycobacterium, which are bacteria that have thicker and waxier cell walls than other bacteria. This waxy coat helps them protect against phagocytes and antibiotics. So we see that the P14 CAN-S conjugate effectively lowered the levels of M. smegmatis to 95%, or by 95%, and lowered M. tuberculosis levels by 93%. And these results show that the P14LR and kanamycin are unable to rid the macrophages from intracellular pathogens on their own, or even when they were co-administered. So the P14 CAN-S takes the advantage of the cell penetrating ability of the P14LRR peptide to co-deliver kanamycin, which effectively reduces the intracellular pathogen levels.